this is exactly the question I'm interested in. How do the cell organize and compartment itself? And this relies on the cytoskeleton, which are the various polymers and tracks in the cell that help it have its shape and move things around so that they are at the right place.
Hello guys, and welcome back to the Neuroscience and Beyond podcast. I'm Svilen, and I'm a molecular and cellular neuroscience PhD student at the University Medical Center Göttingen in Germany. Alongside my colleagues Christina and Sabina, we are on a quest to bring science, and especially neuroscience, closer to the general public. Today, our guest is Dr. Christophe Leterrier, who leads the NeuroCyto lab at Marseille University in France. He is well known for his beautiful work on the architecture of neurons and protein transport inside the cells. And when I say beautiful, I really mean it. Check out the webpage of his lab to see many incredible images of different cell types. To study neuronal organization with nanometer precision, his lab uses advanced microscopy techniques. We're going to discuss all this along with other exciting topics such as science communication, how to build a community on X, formerly known as Twitter, and of course, some challenges in academia. Before we begin, I'd like to mention that this podcast is a separate activity from my position at the University Medical Center Göttingen in Germany. This podcast is supported by the International Max Planck Research School for Neurosciences, as well as the Cluster of Excellence Multiscale Bioimaging in Göttingen. If you also want to support us, please subscribe to our channel and do not hesitate to drop us a comment under our videos. And now, dear listeners, please enjoy the talk with Dr. Christophe Leterrier. Hello, Dr. Leterrier.
Hi.
Thank you for being here. Besides this situation that occurred with the German strike, so the trains now are on strike, the people who are involved there, and unfortunately you cannot catch your train on time. So the people who are involved in the conference yesterday, Encephalon, they were so kind to organize driver for you and drive you to Frankfurt. However, you are still here. And you're willing to discuss science with us, and thank you very much once again for your effort and time.
Thank you for having me. I'm very happy to be here and to have this opportunity to discuss what we're doing. And I particularly appreciate the effort from the German people of having us try it while I'm here. As a French people, that's very dear to my heart. So thank you very much.
We're happy to hear that. Although I'm not German, but still I'm happy for the German people. So usually we start by asking our guests, how did you end in science? So you have engineering degree, right? And also PhD in science. And for me, it's interesting to hear in first place what inspired you to get into the field, and how did you switch from engineering to science, neuroscience in particular?
Okay, so I hope I don't end in science, but this is I don't have an origin story, or when I was a kid I would try to see where the ants would go and things like that. Primarily when I was a kid I was interested in reading and reading everything I could from ads to the encyclopedia. So I guess that's where I got this taste for knowing things and discovering new things. But more specifically, so in France we have this very peculiar system of engineering school, which are a bit of the if you are good in science, this is the best thing you can do, is you do your preparatory classes after the baccalaureate, and then you go to this engineering school, and some of them are very prestigious, and then this is the best way to do if you want to. So that was like the default for someone who was good at school, and so I followed that path. I went to preparatory classes and then to an engineering school. But this is where the things started to veer off the classical path, because the engineering school I went to is called ESPCI, so that would translate to Industrial Physics and Chemistry School in Paris. And it was an engineering school that was extremely heavily oriented toward research. So there was a campus with 15 labs, academic labs in the school, and the director at the time was Pierre-Gilles Degennes, a famous physicist, and really it was one of the only school where you had no management courses and things like that, just hardcore science, quantum mechanics and things like that. And so you were working in the labs. Half of the hours in the school were actually having practical courses in the academic lab. So that's where I discovered really academic research and being in a lab. And I really liked it, and my experience with internships in companies and things like that, because basically half of the students go to R&D in companies and half go to academia. And so I was I liked the academia side more, so that I did a PhD. So fourth year, you can do pretty much what you want, and I did a master's in cell biology. And then there was a neuroscience lab on campus where I did my internship, so I decided to do my PhD in this lab. So that's why I ended up doing a PhD in neuroscience. So that's where I kind of went from classic engineering school career to an academic career. And this is something that I think is important for my way of seeing things, because I am an engineer by trade. So I am interested by things that can be seen as engineering problems also in science.
Mm-hmm. So this is definitely a benefit for you and then for the field, because you then bring some different perspective, and then you can apply it and eventually discover, invent new tools, new things. Maybe we can talk about it later. But you said you decided to go to academia. What made you go into academia, not to industry?
Well, I guess this all boils down to the experience you have, and I went to I made an industrial internship that was in Rhodia at the time. I don't know how it's called now with all the fusions, but so Rhodia had a research center in New Jersey next to Princeton, and so I spent 6 months there in 2000 as an R&D engineer. And the research that was done there, I was very happy to be in the US. It was a great experience, but the research was a bit random. Like I would test everything they had to see if one of those polymers was better to clean cars or bathroom tiles or I don't know what. And this is the third year of the school. So you have 6 months industrial internship and 6 months academic internship. And the other 6 months were in that neurobiology lab, who was headed by Jean Rossier at the time, and that's where I started working with Jolt Lenke, who would become my PhD mentor after that. And I really liked being in a lab and having this, coming every day, discussing about science, what we could do. Jolt was a younger researcher. I was his first student. He was just starting his group, so it was a very exciting time to work, and I really liked that. That we could do anything. Basically, we just have discussing, having good ideas, and do nice things. So that's what drew me to academia.
Yeah, I've heard from a lot of people. For example, some of them drifted a little bit, went to the industry, but then they were like, okay, I earn a lot of money. I'm very happy with my job or whatever, but I lack this push to think about new things and then to push science further and discover and this intelligent thinking about things that nobody else knows. It's kind of something interesting. Of course, it's at the industry as well, but in science it's very flexible and very, very encouraging. You have to do this. This is basically a job, right? And that's why I think it's one of the biggest benefits for me personal to think that we are people who actually basically invent knowledge, right? We write the books and the papers and etc. And it's very interesting to look at that perspective. And now you after this interesting career path, you are leading a department in France, in Marseille, right? Maybe you can tell us more about what is this department doing and where it is located actually.
So, department would be maybe a big word. It's a group. It's a lab.
It's a group, yeah.
So I started my group in 2017. thanks to a funding opportunity. So I am a CNRS researcher. So that's the big science organization for public researcher in France. And I got funding from CNRS to start my group. I was already a permanent researcher at CNRS. I got the position in 2012, and so 5 years after that, I applied and got the funding to start my own group. And so I was already in Marseille because I was working. I did my postdoc in Marseille in the lab of Bénédicte Dargent, and I got a CNRS position in that lab after 5 years of postdoc. Mm-hmm. And then I started my independent group even 5 years later, still in Marseille, but I switched institutes. So because that was also one of the conditions of that funding, is that you had to establish in an institute mm-hmm. where you had no ties to a former mentor. And that's where I started working at this institute that is called the Institute of Neurophysiopathology. Mm-hmm. And we were on the outskirts of Marseille, in the north of Marseille, in a hospital. And in 2019, the institute had a big rearrangement, and we got a lot more groups coming, and now we are in the center city in the Timon Hospital. So this is really an excellent place because it's very dynamic. They are really trying to make a hub for neuroscience there. So there is our institute and a few others around, and it's really there's really a good dynamic. Mm-hmm. So my group is about. So it's called NeuroCyto, neuronal cytoskeleton in health and disease. And we are about 10 people, 10 to 12, depending on exact people that are there. And we are trying to understand the organization of the neuronal cytoskeleton.
Mm-hmm, mm-hmm. Basically. So, okay, now let's dissect these terms, right? For people who have never heard these terms, what is, first of all, cytoskeleton, maybe what type they are, and especially neurons, and why are they important, these things, right?
So I want to say that I am a cell biologist, so I will get technical about cytoskeleton and everything. Sorry for people who wanted to hear about more neuro stuff and cognition and things like that, because we don't look at much more than cells in dishes. So a cell biologist that studies a cell that is a neuron. So that's how I would define my relationship to neuroscience. And so neurons are incredible cells, beautiful and incredible because they have these intricate shapes, and they are there's not a lot of human cells that can be 1 meter long, and neurons are these. So basically, it's an extreme example of how cells can take very weird shapes, and this poses a lot of challenges in terms of morphogenesis. How can you build this shape, and once you have built it, how can you put things at the right place and have the protein being sent to a very distant place in the cells and compartments that are very well defined and very different from one another. So there's this is exactly the question I'm interested in. How do the cell organize and compartment itself? And this relies on the cytoskeleton, which are the various polymers and tracks in the cell that help it have its shape and move things around so that they are at the right place.
Mm-hmm. For the sake of definitions, can we maybe say that there are several these microtubules, right?
Yeah.
The actins or the neurofilament, right?
So the classical cytoskeleton elements are defined as three types. So microtubules that are made from tubulin, which are big cables that are mostly for transport on things, the vesicle that are traveling along. Actin, so actin makes filaments, and actin usually has more like a role for shaping the cell, so it supports the membrane and helps the cell not being just a big bag where everything is mixed. And neurofilaments or intermediate filaments, which are like the a bit overlooked usually, but which are very important also for structural support of the cell. And some people could argue that you could add stuff like spectrins and septins and all kinds of other polymeric proteins, but I would say these are the three main types of cytoskeletal proteins.
Is there any explanation why do only like microtubules are involved more in trafficking than actin, or does actin also play a role in trafficking? And under trafficking, I mean the things that you just said, that some proteins, let's say in the cell body of the neuron, have to go to the very long distance, travel along the axons, which are this nice, beautiful long protrusions, and go to the synapses, for example, and they need to be trafficked. And along this cytoskeleton molecules, we have trafficking. But why is it like one type is only for this? Is there an explanation about that?
Well, of course, it's of course an oversimplification, because there's a lot of cases and cell and neuronal types, but I would say the way I see it, at least, is the microtubules are more like the stable tracks that can go long distance in the cell. And actin, you cannot have an actin filament that is like 50 micrometers long. So you would have to have something more complicated with several filaments, and you have to assemble, and that will be a bit flimsy. So microtubules are more adapted to long-range transport. That doesn't mean that actin doesn't have a role in transport, but it's less the role as the roads, but the role as regulators. Mm-hmm. So you can imagine that you have a microtubule and something traveling along the microtubule, and then you have an actin assembly that is there and that stops the vesicle when they pass and decides should it go back or go forward or be exocytosed there. So the regulation of the trafficking at short range can be done by actin. So this is, I would say, the main way to see it is microtubule for long range, and short range regulation would be done by actin assemblies.
Mm-hmm. So the heavy logistics, so to say, is done by actin, and microtubules are the highways that you can send.
Exactly. I have a slide like this where you have like the microtubule in the axon, and it's a highway, and you have the exits where you have actin assemblies that help direct things like a toll on the highway to the right place.
It is very nice when you fly in the evening, for example, over some big city, and you see these lights, and then the roads that are like all with lights, and you see the cars traffic, and sometimes I imagine neurons, that you have the concentration of things in the summer in the city, and then the trafficking all around the roads. It's very beautiful. And you are focusing now, or at least to what we heard yesterday, once again, for the people who are not aware of, yesterday there was a very nice symposium in Göttingen called Encephalon, organized by the local students from the CMPB program. And we heard very nice, inspiring talks, and you showed us very nice data. I don't know if we're going to discuss this on the podcast, since this is unpublished and people did not see it, but you studied there the organization of a particular part of the axon where actually the action potential is generated. Maybe if we talk a little bit about what action potential is, and why is it generated exactly there, and what is the importance of this part?
Yeah. So we studied the neuronal cytoskeleton, and in particular the axonal cytoskeleton. So as you mentioned, this very long extension that connects to other cells, and there was a lot of I would say our view of the way the axonal cytoskeleton is organized has been completely transformed in the last 10 years. So besides microtubule highways, we know that there are lots of things we might talk about, like the actin rings, periodic scaffold under the plasma membrane, and all kinds of strange cytoskeletal assemblies that are really unique to this compartment. And so we basically, that's where we work. Like, we would try to understand and make sense of all these things that have been discovered not so long ago. And so the action potential is, of course, one of the defining process that occurs along the axon. So that's the generation of an electric impulse that travels along the axon and triggers the release of the synaptic vesicles to contact and to connect with other cells. And it is made at the beginning of the axon, at a particular compartment that's called the axon initial segment. And this is something I've been studying for a while, because that was like the subject of Benedict Dargent's lab when I came for my postdoc there, and the way the action potential is generated is because you have a concentration of sodium channels, so specific channels that can transfer ions from the outside to the inside and back, so that you can generate electrical potential differences that will make the electrical impulse. And it is very interesting because it's a case of people started with a more electrophysiological channel aspect, and when I came in Benedict's lab, it was really about the ion channels. Where are they? What are their characteristics? But if you want to build something like that, you have to have a kind of complex and a kind of structure. And this is what I was interested in. Like, what are the cytoskeletal structure and the protein that can anchor them at that place? And this is where the subject drifted from ion channels to more cytoskeleton. And now the recent work we have done on this is less about the fact that it's where the action potential is generated, but it's more of a technical, I would say, a constraint, because we are working with an electron microscopist who's called Stefan Vassilopoulos, and we are using unroofing. So we are opening the cell mechanically to see what is inside by platinum replica electron microscopy. And this is much easier to open the beginning of the axon than further when it becomes a very thin tube. But so that is one of the reasons we are still working on that part of the axon. And there is, of course, a lot of very interesting things happening there, as you mentioned.
Can we maybe clarify something? So for us, is interesting. Basic science is very interesting, right? But for people who are not familiar, it's like they might ask, why do you study all this? What is the relevance? And maybe if there is something disease-related, or I'm sure there is, but maybe you can elaborate on this question.
Yes. So my institute is the primary I would say relevance in pathology, and the type of pathologies that we are interested in studying and understanding are neurodegenerative diseases, and for the most part Alzheimer's disease. So we are not, our group, we are not directly looking at disease models or things like that, but we are always we always pay attention to the potential role of of cellular processes we're studying in pathological contexts. And of course, the axon, for example, as it is a very thin tube, very long, is one of the first structure that is degenerating when you have any kind of neurodegenerative disease. And of course, if you lose this cable that connects the neurons, that you already start to have a lot of problems, even if the neurons themselves are still alive. And so this is and of course the structural support is one of the first things that can go wrong and that can cause the axon to degenerate. So I said there are several steps between what we are doing and the actual pathological relevance, and I don't want to claim that we are covering all these, but we still always have this this in mind, like, oh, okay, maybe this structure we are looking at, it might be perturbed in the disease, and if we have the occasion or the opportunity to have either simple cellular models, or I have the lab just next to mine is from Emmanuel Nivet, is using iPSCs, so these induced pluripotent stem cells, where you can make neurons from human people and from patients. So there you can have cellular models and neurons from actual Alzheimer's disease patients. So this is something we try to go toward, to look at really fundamental cell biology, but in models that are very relevant to pathology.
That is true, and this is why it's called fundamental science, basic science. We basically push the things that we still don't know in order to understand, right? And it takes a lot of time and effort, and sometimes it does not lead directly, as I mentioned, to a cure of some disease, but brick by brick we pave the way, so to say, eventually in the future.
As I often say, we are looking at how things work to understand how they don't work. So.
That is true. That is good analogy. So going back to your research, it's a nice and good point now to clarify what kind of model organism do you use to study these things?
So I am using a model of cultured neurons. So neurons cultured on the glass cover slips. And those neurons come from rats. So embryonic rats, not too long before birth. And we isolate this region of the brain called the hippocampus. So the full name would be embryonic hippocampal neuronal culture from rats. And the reason we use this model is because it's a classical model that has been devised by Gary Bunker at the end of the 1970s. Mm-hmm. And that is one of the most detailed models because you can get beautiful neurons that are extremely sparse. So you can really know, you find an axon and you can follow it all the way back to the cell body and you can really do subcellular work. And so this is the primary reason why we use this model is that if you take a brain or a mixed culture, you know, it's super dense and the cells are all over each other. So if you make a simple staining, you don't know what you're looking at. You know, is this dendrite belonging to that neuron or that neuron? And here you can really spread them and have them very sparse. So you see exactly with your microscope, you know, where are the various compartments and make live cell imaging. And that's the primary reason. And as I said, for me, these are the most beautiful cells. So this I would say the aesthetic aspect is also important.
I also work with primary hippocampal cultures, and indeed they're fascinating. I assume you don't have only neurons in your culture, but also astrocytes, the supporting cells.
So we use this. I mentioned Gary Bunker, and I think the type of culture is called Bunker type because he pioneered the fact that we don't culture the glias and the neurons together, but we culture the glias on the cover slips and the neurons on another cover slip, and then we make a kind of sandwich. So we can have a very low density of neurons on the cover slip with the neurons, but the astrocytes on the facing cover slip are feeding them. So we can, we try to minimize the amount of cover slips we have on the cover slip itself, but this is a sandwich co-culture.
Yeah, we had this exact culture also, but due to the restrictions that are coming with animal experiments, we had to stop them. But this is completely other topic right now.
This is one of the reasons we are interested in these human pluripotent stem cells also, because they are easier to use in that context.
Yeah, but they're still not at all that well developed, but we had a podcast on the topic. Please check it out with Patapias Afero. Yeah. So we had a brief discussion while we were walking towards our institute and talking about why it's important to have this culture as a model and what is the relevance for the physiology. Maybe if you can elaborate this.
Yeah, I mentioned that because that's something that's very important. And when you are doing cell biology of neurons, you always have these questions from everyone basically, but neuroscientists in particular. Okay, but these are very artificial cells on glass. You know, is it really relevant? Does it really happen in a real brain, in a real animal? So when will you start doing that on slices or on in vivo and things like that? And I think the logical or the political answer would be, we want to do that. It's very difficult, and actually not. We don't plan to do that. And now I'm more like fighting back a bit and saying, no, we are using a very artificial model. But this is the essence of what a reductionist approach is and a modeling approach is. So I usually answer now, I know a lot of things that happen in slices that don't happen in my cultures. I don't know a single thing in my culture that doesn't happen in the slices. So we are basically simplifying and purifying the process. And what remains in those very artificial cells on cover slips is likely to be very important because even something as traumatizing and artificial, like putting them on glass, doesn't remove those very fundamental mechanisms we are looking at. So I try to, turn the tables on the argument and say, yes, we are doing something very different, but this is basically the core of what is important at the cellular level that remains.
I've never thought about this, but I think if you allow me, I will pick up this argument and then I will use it since we also work with cultures. And it's very, very important to keep in mind this. So now we established you're studying this interesting highways in neurons, and you're using cell cultures to do that. But also you use a very nice techniques, which I am very happy to discuss with you about, microscopes, and in particular super resolution microscopy. So now you said that your collaborator, you had this electron microscopy things going on. Let's maybe start talking about what is super resolution? What is resolution at all? And why do we have to invent some methods to overcome some limitations, perhaps? And since you are very familiar with this and have a lot of experience, I would like to hear from you about these things.
I hope I'll do a good job. I mean, there's lots of people explaining that, and I think there's a lot of very good explanation. I hope I can do a good job. Light microscopy is limited in the resolution you can get. So if you make an image of a cell with a microscope, you magnify stuff and you look at the image you get. And the intuitive things would be that, okay, if I want to have more detail, I can just magnify more. And I will get something that is, and I will see something that is smaller. And it doesn't work like that because fundamentally, you don't get all the information from your sample with a microscope. And this information that is missing, it's an information at the level of the object you are looking at. It's an information that is spatial, and the translation of that in the spatial realm of your image is a level of detail. So it's an information content problem at the front. I hope the physicists will not say that I'm just saying wrong things, but that's the way I understand it. Like, you cannot get all the light that comes out of your sample because you only have a limited amount from the point you are looking from, and you don't see, because of course your object has light that is emitted in the case of fluorescence in all directions, and you are looking at a small portion of it from a certain angle. Mm-hmm. And all this missing light forbids you to be able to refocus the image to an infinitely small detail. So if you have more, if you manage to get more light from your sample, but you've put other objectives, for example, you will be able to refocus a smaller object. So this lack of that you don't get all the information from your sample translates into the fact that your sample will, if it's a very small point, will be a point that is a blur that is a bit larger. Mm-hmm. So this is a fundamental property, and you cannot do much about it. You can add other objectives or objectives that are huge, and so this is what is called numerical aperture. Basically, it's like getting more light from your sample, so you can refocus it more precisely. But there is a very simple, actually, relationship. If the amount of the proportion of the field of the light emitted you can get, the wavelengths of the light that is emitted will define the size of your spot. And this is called the point spread function. And if you want to bypass that, you actually cannot break. So when the super resolution optical microscopy was invented and started being used, there was a lot of people saying you break the diffraction barrier, because this phenomenon of not being able to refocus is called diffraction. So the diffraction barrier, and it's not being broken. You never break the diffraction barrier. You find kind of sneaky ways to bypass it, but you never really. This is a fundamental physical constraint. So there have been a lot of different techniques that have been invented in the last 20 years that try to bypass or to play with this limit. And I would say the overarching concept is that you have to modulate the light that is emitted from the sample, and so you make it in some way blink. You have as Stefan Hell was saying at the symposium yesterday, you have to have an on-off system, and if you can drive your sample to have some fluorophores that are on and some fluorophores that are off, you can get more information, and you can somehow get this missing information to get a more precise image.
And to the people who don't know, fluorophores are these probes that we put on cells, on proteins to mark them, and when we shine light to them, they emit light eventually, and we can detect this with our detectors on a microscope or cameras, right?
Yes.
And in principle, people use the so-called epifluorescence or confocal microscope, which has certain limitation, and we can see up to, let's say, roughly 200 nanometers in the lateral resolution, XY planes, right? And the axial resolution, or the Z plane, is a little bit lower. This is again because of the properties of the objective lenses and light, and we cannot overcome these limitations. But now we have this kind of a super resolution microscopy, and for example, the STET, which was invented, one of the people, Stefan Hell, the Nobel Prize laureate, right? Then we have the structured illumination microscopy, or STORM and PALM, and all of these other terms. Maybe if you can explain one of these, pick up your favorite, for example. I know you work with SIM a lot, and also you work with STORM.
Yeah.
But I don't know, for the actin rings and the actual
Yes.
the initial segment, actually, Zhou Zhang, right, from
Yes.
MIT, she started with these STORM images. They were very beautiful, right?
So yeah, maybe we can start with STORM. So STORM stands for Stochastic Optical Reconstruction Microscopy, and the acronym was coined by Zhao Wang from Harvard. And there is a host of these techniques that have a similar principle. So STORM is one. There is PALM for photoactivation localization microscopy, and PAINT now. So I would say that if you want to have a general term, this is what we call SMLM, single molecule localization microscopy. So it's really an acronym soup. I apologize to the to the people listening. So single molecule localization microscopy, the name tells you about the technique. It's the idea that instead of looking at all your fluorescent emitters, the fluorophores you explain, at once, because they have, each of them will make this kind of blurry spot, and all the blurry spots will be superimposed, your image will be a bit blurry. But you cannot resolve them because they are overlaid. And the idea of single molecule localization microscopy is that you will put all of them to a dark state, no emission, and only a few of them will emit. So you will see what I call the point spread function. So this point that is made by a very small emitter. And as you see the round shape of this spot, you can pinpoint precisely the center. So you can locate the molecule with a much better resolution than the size of the spot it makes on your image. But you have to be sure that it's a single molecule and that it's not an overlay of several of them. So what you want to get is a blinking sample where everything is dark and a few of the molecules are on, emitting light. So you have just a few spots, and you pinpoint their position by fitting very precisely their center. And then they go off and others go on, and you pinpoint a few of them. And maybe you have 10 of these on every frame of your image, and you need 50,000 of those frames. So at the end, you have 500,000 of these spots that are precisely localized, and you make the image, and the image you make is basically a graph of all the positions you have fitted of these 500,000 fluorophores, and that image will be much more resolved than your single snapshot of the fluorescence. But it will take time because you will have to take 50,000 frames of blinking fluorophore. So basically you are trading spatial resolution against temporal resolution. So you just spread your sample and your labeling along time. So you build it progressively point by point. This is why it's also called pointillist microscopy, referring to the painters of the 19th century who would paint by doing little dots on the canvas.
And at the end, when you acquire all this information, there's this heavy computation that allows you to actually localize where in the space X, Y, Z these molecules are located, and then you kind of make your image out of this. It's basically also computation involved. We will touch upon this a little bit later. But I wanted to ask, okay, now you actually said you trade temporal for spatial resolution, right? So perhaps this method is not suitable for live imaging?
Yes, that's true because if you need 50,000 frames to make one image, even if you go very fast, you can imagine. I mean, typically for us, and we are more optimized for qualities and speed, but it takes about 15 minutes. So of course, if you want and you can get to a resolution of about 20 nanometers, you can imagine that if you have an image of 20 nanometers resolution, that takes 15 minutes. If you do that for a living cell, the cell would be at a completely different places 15 minutes later. So your 20 nanometer resolution is useless. Mm-hmm. So that's a lot more adapted to fixed cells. And for living cells, what we have been using for a while now, a couple of years, is structured illumination microscopy that you mentioned, which is a very different technique, but fundamentally, you are structuring the light that is emitted. So it's the same concept of having an on-off contrast to gain resolution. But here what you do is that you project grids of illumination on your sample. So that's fringes with different orientation and phases. And if you have 9 of these phases and an orientation combination, you can look at the moire effect that your sample is doing with these fringes, and you can basically I would say intuitively what you can say is that you can infer about the smaller details from the larger moire that you see. It's difficult without an image to explain, but you can infer mathematically. And this, some people don't want to call this super resolution because it's actually the optimal use of classical microscopy. Mm-hmm. And the limit is really 2 times the diffraction. Mm-hmm. So you can go from 200 to 100, I'd say 120 nanometers. But some people say this is not true super resolution because there you are still within the realm of the diffraction limit. You are just optimizing the way you illuminate so you can really get all the information. Mm-hmm. But the advantage is that you just need to do 9 images in case of 2-dimension SIM, and 9 images for every time point. That's something you can do in less than a second or 100 milliseconds. So you can do snapshots and make reconstructions.
I remember there was a claim from Zeiss, one of the microscopy company, that you can go up to 70 nanometers, 60 nanometers with SIM squared, I think.
Yeah. So this was, as you said. There is a lot of computation involved in all these techniques, and SIM is not, of course, is one of the techniques where you have a mathematical framework to be able to, from the 9 images that are modulated by your fringes, you infer a final image that has a 2 times better resolution. But the idea is that if on top of that you introduce some information, so you deconvolve. On top or before you are doing the actual SIM reconstruction, you deconvolve your images by basically injecting information like the sample, the fluorescence is sparse because the objects are less than 1% of the image. The sample should be continuous, like it cannot be completely random pixels. So you are using the information you have on the sample to kind of guess and get a better detail by deconvolving and then applying the SIM. Some people claim that you can get a better resolution. Mm-hmm. What I think is that what you get is a better contrast. So if you are lucky and your sample is adapted to that basically thinning mechanism, you will get something better. But if your sample is actually more complicated than that and your assumption when you deconvolve are not entirely true, then you get things that are more artifactual. Mm-hmm. So you have to be careful with those added mathematical steps and the claim of resolution they are associated with.
Mm-hmm. Deconvolution, convolution, these are mathematical things that we're not going to discuss now. They are complex for the listeners, also for me personally. There is also Fourier transformation and Fourier space, etc., which is actually quite important in signal processing and also in imaging. And for everybody who wants to go to the imaging field, please keep in mind these things. And this is the future, mathematics, programming and computation and microscopy. But I still did not get why is it SIM or the structural illumination microscopy better for live imaging than
because it's much faster. Mm-hmm. And it's a wide field method. So you basically just do 9 images and then you have your reconstruction and your image. Mm-hmm. And it doesn't need a lot of light. Whereas STORM the way you get those, in addition to being very slow, the way you get your sample to be mostly dark with just a few active is by shining a lot of light. So you have a very high laser power. So in addition to being slow, it's extremely damaging to living cells. Whereas SIM, you can do it with the same kind of intensity you would shine with a regular epifluorescence microscope. Of course, if you have to take 9 images to get one, that means your photon budget is 9 times higher. So if you are already at the limit with classical imaging, then if you want to do SIM, your cell will not like it and it will die. You will have photobleaching, phototoxicity. And this is definitely a challenge that we are trying to address, in particular in neurons, which are very fragile cells and cells that don't like light, very sensitive to photo damage. So you have to really go lower and lower in the power. And the result of that is that when you want to process those SIM images with your mathematical algorithm, you get artifacts because your starting image is very noisy. And so this is the challenge. Like, you have a technique that is quite reliable, but you kind of push it to the limit because you want to do it with as little light as possible, but then you have to find a good balance, because if you go too low, then you have too much noise, and you start, basically your algorithm will start add a lot of details that don't exist because it doesn't know what the image looked like in the beginning. So this is why we are also very interested in what we can do as a later step with all these algorithms of denoising and deep learning based that can help us retrieve or enhance the image we have by training a model that has seen good and bad images and is able to transform bad images into good images, basically.
Maybe now we can drift away a little bit. So far we discussed techniques that are based on the microscope, right? Storm, and also on the fluorophores. Storm, STET, Palm, SIM, etc. But now, in the past, 10, 15 years, there were some methods emerging that rely only on computational processes to get the information that we want based on this point spread function, etc., and use a normal wide field or epifluorescence microscope, and from the images that we acquire to get a super resolution. Jörg Enderlein is here in Göttingen. He is one of the pioneers, and SOPHY it's called. I always forget what the acronym stands for. It's a fluctuations. Then we have Ricardo Henriquez and SRRF, and we also use it in our lab. We are heavily involved in expansion microscopy, which is a different method to basically get more information and not relying on the machine, on the microscope. Can we maybe start talking about this? How can we get information in the nanoscale out of normal images without these machines?
Well, I would say that's an even more technical and mathematical discussion. But and I'm not sure I could actually explain very well on that front. But yeah, so we are using SRRF, and we are lucky to collaborate with Ricardo Henriquez in Lisbon, long-term collaborator. So we were involved in developing the new version of SRRF that was just out, and that is really easier to use and more robust so that people that just wants to have a better resolution don't risk to have things that are full of artifacts or things like that. And the principle of this fluctuation-based technique like SOPHY and SRRF are in a way similar to SM2 single molecule localization microscopy, but instead of really getting them on and off, they are just fluctuating, and they stay so the fluorophores stay overlapped to each other. But by looking closely at the pattern of the fluctuation, you can kind of say, oh, okay, the fluctuation, the variance of the fluorescence is higher here, so maybe we are in between two fluorophores, whereas here it's more, so there is a fluorophore here. And the SRRF in particular is based on radial fluctuation. So the idea is to kind of push the light back to its to where it was emitted by looking at the fluctuation. I won't go further into the mathematical details, but in practice, basically this allows you to this can be seen as a spectrum from completely continuous imaging, wide field, to single molecule localization microscopy. And if you are in between, where you have things that are fluctuating, basically you don't need 50,000 frames like single molecule localization microscopy. You can do just 1,000 frame of fluctuation, and you will get an image that has, I don't know, maybe 80 or 100 nanometer resolution. So it's kind of halfway between wide field and single molecule localization microscopy. And as you said, the advantage that you just need to make a movie of your sample that is fluctuating, and you can do that with a classical microscope.
And this is like opening up the field, right, to people who cannot afford these expensive instruments. Most of them are up to 1 million, even more, for a microscope at the SIM.
I want to emphasize that if this is possible now for everyone to use these techniques, this is because Ricardo and Guillaume Jacquemet in Finland are putting a lot of efforts to bring all these techniques in very simple software and open source software. And so I think this is something that I want to stress, that, it's not only the work is not only to make the algorithm, but to make sure that they can be used by anyone. And you have to really, I just wanted to stress that point.
How does it work? Do you have a plugin for, we as imaging people, we use a lot ImageJ among Python and other the software, but do you have like plugins and they're somewhere online? How does it work?
You mean
the SRRF, for example, the enhanced?
The NSRF is really integrated as a plugin, like the first version. And there are also, because now Ricardo's lab is also shifting to Python and Napari, so there are also plugins for Napari that can do most of what the ImageJ algorithms can do. So the idea is really to identify what is the most used solution and to try to fit that, rather than force people and saying, no, we as coders think that this is better, so you have to learn that to use it. So this is really going the extra mile to put it in the hands of the users.
This is remarkable, and shout out to these people once again. Maybe at some point we can invite them in Göttingen and have them on the podcast. It would be a pleasure. So, okay, now you have these super resolution methods and also computational methods. And you mentioned that in your research, or in your latest research, you use the electron microscope, right? So how do you combine the benefits of both realms, for example? So
I'm able to use electron microscopy because I have this collaboration with Stéphane Vassilopoulos, who is really a gifted electron microscopist at the Institute of Biology in Paris. And I have to say I've never done that myself. But this is the core of the kind of work we do, and what I like. Because, I don't code algorithms, but I have this wonderful relationship with Ricardo, and I am able to work with him. I don't do electron microscopy myself, but I can work with Stéphane. And I think this is, we were talking about what is good in academia. That's something, if you're not inside the company, there's no way you can go to work with someone else, because just the technique they use is incredible. So this is one of the perks of academia. And how do we combine this? The idea is that so this technique that Stéphane is using, platinum replica electron microscopy, is really adapted to samples on glass, because you are unroofing them. You just keep the part that is stuck on the glass, the ventral membrane of the cell, and then you make a metal replica of that, and this is what you look at with the electron microscope. And so this gives you an incredibly detailed and contrasted image of everything that is linked to the membrane. And so that's why we wanted to use it to look at these actin rings that are just under the plasma membrane along the axon. And we figured that would be the way to finally see these actin rings by electron microscopy, which was never done since Richard Weiswang discovered them using STORM. And so we could do this. We could unroof the neuron, and Stéphane could do these magnificent images where we could see these actin rings and determine that they were actually braids of two actin filaments braided together and very long, which was a surprise given what was expected. But then once we had this, we'd say, okay, we have the super resolution image where we see these regular bands, and we have the prime image where we see these little braids. But can we really make sure these are the same? And so so what we did was to try to actually do a storm image and a prem image of the same exact sample, like correlative storm and prem. We were not the first to do it. Justin Tarasca in the US has been pioneering the approach where you do a super resolution microscopy and prem and then correlate them together. So, we said, oh, it must be very difficult, two very high-end techniques and bringing them together. And actually it was, once you have them separate, it's not that hard to put them together. And the fact that both techniques are super high resolution actually makes it easier to superimpose them because there's no ambiguity. If you try to superimpose a classical fluorescence image on an electron microscopy image, you end up with these giant blobs of light, and they could be almost anywhere. So you have to have fiducials to make sure where you want to align them. Here, the storm image, a cluster of fluorescence on the storm image, is basically coming from one antibody. So, and you see that antibody on the prime replica. So you can basically just put the cluster on the antibody, and when it's really well aligned, visually it kind of locks in, and you're like, okay, that's aligned. There's no ambiguity. So you don't need fiducials. You just need like a basic software. You have the landmarks, and you say this goes here, and then bam, it's correlated. So it's a lot of work, of course, but it was not as difficult as we thought it would be.
And in the result is remarkable. We saw some of the images. So guys, Dr. Letterrier. Sorry. He's very active on social media and posts a lot of beautiful images there. Oh, yeah. So go ahead, check his X now account, not Twitter, and you can maybe also see these beautiful images. Maybe now we can touch upon this. How do you see, you're quite active on this social media, right? How do you see this as a person involved in science? Do you feel, okay, I need to communicate my research, or you just do it for fun? Or how does it work?
Well, I think the way I started using social media and Twitter in particular was really just like there was no predetermined strategy or things like I need to make my science known or things like that. I think it was just like I was really, I've always been very interested in discussing and seeing what others are doing. And I cannot be happy just doing my science in my lab alone. And, some people are very happy like this. I need to really, feel like I'm part of something bigger and part of a community. And so Twitter was a way to actually meet and make that community, like, even before you people know who you are and invite you to things and you go to meetings. This is like a worldwide meeting all year long. So and it has been tremendously useful and beneficial for me and for my science because it's as you said that a lot of people know me from the communications and my presence on social media, but I have to say that it's not. I mean, I think if you do it as a strategy, like okay, I have to advertise myself, it will not work as well. Like it's really like, oh, there are these good people, I will chat with them, and it takes years. Like it's not like you can, just use Twitter as an output where say, oh, look at my paper, look at my images, because you have plenty of people doing that, but that's not what's interesting. What's interesting is you bring original content that people like, and you build a network, and you are part of a network. And there's this paradox because social media is often seen as something very instantaneous. But building a real network of people that care about what you have to say and you care about what they want to say takes years. Every day, maybe it's 10 minutes, maybe it's 1 hour, maybe it's 5 hours if you're really incapable of logging off. But you do that every day for years and years, and in the end, without realizing it or having a strategy, you realize that if you have a question, the first place you go to have an answer about anything like a protocol or a paper that would look at that or this is your Twitter account and all the people you interact with. So that's something very organic, I would say.
It's remarkable how easy it is to reach out to people that are on the other end of the world without even putting effort, just tweeting something and eventually someone retweets it, somebody else sees it. You even don't know the opportunities that can be created, right? And this is like fascinating for me.
And so I just want to add another point is that people often see that as virtual, like, it's virtual life, it is disconnected from real life. But for me, I had plenty of opportunities in real life and in my actual research that came out from interaction I had on Twitter, collaborations with scientists, invitation to seminars and things like that. So, and I remember the first time, I think that was maybe in 2014 or 15, and I asked on Twitter because I was looking for one of these new red fluorescent protein. I think it was tandem tomato or something like that. And someone replied to me, said, "Oh, I have this plasmid. I'll send it to you." That was Steve Royle in UK. And 3 days later, I received, and for me, it was magical. I mean, Agin didn't exist. You had to write to someone, and most of the time they wouldn't answer. And now I just say, "Does someone has this red fluorescent protein?" And 3 days later, I have it in my mail, and I was like, "This is incredible."
yeah. See, but of course, it's double-edged sword, right? I mean, for people who are aware of the danger of this social media and that you can go to the rabbit hole and lose your time and start thinking about posts and likes, of course, this is very dangerous. But for someone who knows how to use this tool, because it's at the end is a tool, then it's quite beneficial, right?
There's plenty of ways to do it, and my approach was always to, I have no strategy. I never say, "Oh, I should post this or that," or "I haven't been here on Twitter today, so I should spend 10 minutes." No, just I do it as I feel it, and it works well. So I'm lucky for that. But yeah, I mean, there's plenty of ways. Some people are more reflexive about the way they do it, and that's fine.
Mm-hmm. Yeah, at the end, everybody can think of him, herself, themselves, or whatever. So they can decide, right?
And I agree that there's the danger is that the dark side of Twitter and X now even more is this polarization and fight and things like that, and political divide and I would say the reverberation of the conflicts in the world, and so it's sometimes, most of the time we just don't talk about that with our, we are academics, we talk about things that are much less tragical, but sometimes you cannot do it because the what happens is just so important, and so sometimes you have to say, okay, let's just forget about Twitter for a few days, and and we'll see when I'm back.
Yeah, that's true. But honestly, I like it now when it's so raw somehow, because you can now really see the real behavior of people doing things that, I don't know, previously maybe they would have not done it. And it's interesting to follow some dynamics. It's personal opinion, right? So, but yeah, I totally agree. And my personal X account is full of science. Then you have war, politics, science, and it's a huge contrast. Yeah.
I have cooking on my account.
You like cooking?
I like it a lot. It has replaced bench work.
How did you find German food here?
I had good food. Okay. I will stay diplomatic.
Okay, that's a good answer. Yeah. Cooking is exactly like doing some protocol.
No,
right?
I realized. I mean, there wasn't like a conscious strategy, but I realized I cooked more and more as I was doing less and less bench work. So I think it really replaced being at the bench.
Okay. Cakes are also something like this. Exactly the same recipes. Yeah. Okay. So I think now is the time to drift away from the positive things in science and go to the dark side, if you want to call it. So towards the end of our podcast, we would like to hear opinions from scientists about the problems in academia. And there are plenty, as any other field, but usually we try to gravitate towards reproducibility of your experiments, or people who are well trained and leave academia because of the harsh environment. Also, the publishing industry is not the well structured. So there are many others, of course, the funding, how you get grants, the working environment, if you wish, and other things. But if you maybe pick up some of these aspects and comment on what would be your take? Okay, this is wrong, and I wish this would have been done this way, and maybe give us advice as we are like young scientists who eventually will stay in academia. What can we change to be a better place, right?
I think that's, I wouldn't say negative, because these are opportunities for change and I think there's a lot of things that are happening right now that slowly make things better. So I would have an optimistic view of the negatives, if you want. So about the publishing. So maybe I just want to say a few things about working in academia and the fact that it's a hard. It's an occupation that is hard because you are constantly. Basically, doing science is failing 95% of the time, and this is at every level. Your experiment never work, your funding application never work, your papers are rejected. So we are living in a world of failure, and this needs a very specific mind frame to not become bitter and to actually thrive. And I think the most successful scientists I know are the people that get every of these opportunity to learn and to be better and to make things a bit better the next time, and seeing failure not as failure but failure as just opportunities to become a bit better. And if you keep this mind frame, I mean, basically you will get better and better and you will succeed, but we will never succeed all the time. You know, it will stay, and if you don't like to not get what you want 95% of the time, that's not science and academic science. The career you should follow. That's for people that really like to do hard stuff and on the long term. So another thing is that the main quality for me of a scientist is persistence. It never works for the first time. It never works for the second time or third time. Maybe it will work the 10th time. And that's true for your experiments, and that's true for your funding application. And, you know, some people are extremely lucky and everything works for the first time for them. And then one day it will start being back to the normal way science works, and they will be very surprised when that happens. For me, I was lucky that I started with a lot of failures, like my 10 or 15 first grant applications were rejected. So when one worked, I was like, oh, that can happen. Great. And now, you know, when I have like only 3 to 4 failures in a row, I'm like, oh, that's good. so yeah, no, this is something really that people should keep in mind. And of course, people will talk about the successes, the grants they have, the paper they publish, but we are all just, you know, but yeah, like I don't know, if you're a climber, you don't want something easy. You want to, you know, you want something that is hard to climb. So science is the same. It's hard, and so it's mostly made of failure. So it's just an opportunity to do things better. Now about the I would say the organization of science and the publishing, for example, I'm very optimistic right now, because we are witnessing a profound transformation of the way the science is communicated and is published. And there are lots of experiments. Some of them will work out, some of them will not work out, some of them will actually be maybe worse than the system we have. But it's very good because there are a lot of problems with the way what is strange that people say the way science was, but the review, the journals with peer-reviewed paper is a very recent invention, actually. I mean, it's from the 1960s. So this was not the way science was published before that, and it's not like the definitive way it should be. So it is a system that has its advantages and disadvantages. And of course, the thing is that we as scientists and as academic researcher, our currency is recognition. So everything, every system, every way you can set up the publishing system or the way you communicate your science will be gamed by the maximization of recognition, because that's you know the incentive we all have. So you have to be careful about this, but I think there are ways to make things better. And of course, I would say the preprints are the most important change in biology, in biomedical science in the last 10 years. We are celebrating the 10th anniversary of bioRxiv, and I am a bioRxiv affiliate, so I'm part of the team that screens the preprints before they go online to make sure there's not a preprint that proves the existence of God or I don't know what. So we are not reviewing it. We are just checking that it looks like a regular article for 30 seconds, and then it can go online. So the good thing is that we are involved in the life of bioRxiv and the way they are deciding to change things and everything. And I think this is an extremely important and seismic change, like in the way, and it has all kinds of consequences downstream. It doesn't mean we will not have journals or not have peer review, but it means that we will do it differently. And as was said by the bioRxiv founders, you uncouple making your science available and deciding what is its quality. So now it's two different steps. You can make it available and everyone can see it, and then a journal can put a stamp and say this is a good paper. It has been reviewed by 3 anonymous experts, or someone can just look at it in a recruitment committee and say this is very good science. I don't need 3 reviewers to tell me that this is a good paper. So then you put back the decision to make your science available in the author's hands, and then you have lots of different validation that can happen afterward.
What do you think about this funky idea? Let's say AI, the artificial intelligence, developed to a certain point where the community creates some algorithms that will base your application on the design, on the data, on the language if you want. And then you get a score. You put it on bioRxiv, you get a score, and then you don't do peer review, for example. Could this be something like done in the future?
I'm sure it could be done. I'm not sure that would be a good thing. I think, as I said, any system you put in place will be gamed because there are bad actors and good actors, but even the good people are the incentives. If you know that there will be a score associated with your paper and that your goal is to maximize this score. The saying, like every metric becomes every measure becomes a target. So people will start optimizing their paper for that. And I don't think AI is at the point where it's for now. I think it's too easy to game. So if you start having that kind of system, people will discover the way to make sure they have a 9.9 score every time, and it will be useless. I mean, there might be possibilities to make it to make something that is more automated than having actual human peer reviewers, but. First, I don't think it will replace them. That will be always, the two will be necessary. And second, it has a long way to go before it's an easy target for gaming.
Okay, I might, again, torture you a little bit on the publishing. So 3 things, for example, the reviewers, they don't get paid for their work.
Yeah.
Also, it's officially anonymous, but when you revise a paper, you can see that people tend to cite their work, and you can easily find out in most of the times who is this work and from, for example. This might ignite some conflicts of interest, right? And how could you overcome this? There's gonna be always this human bias, but how can we do something about it to make it as objective as possible, right?
So what was your first?
The payment of the reviewers.
Oh, yeah.
They do it for free, basically, right?
I think both of these things, like the fact that you work for free and the fact that you are anonymous, are important. And I think it's possible to imagine a system that is different, but I think the disadvantages will overweight the advantages. So payment, I'm not sure. I think people don't realize that it's actually expensive to make a good journal, and that would add to the cost. And I think, I mean, the problem is that there's just too many articles. It's not that we are not paid to review them. It's just that the workload of reviewing these millions of papers has become unmanageable. But that's mostly a problem with the fact that we are all trying to churn out too many papers because that's the currency today. So I say it's more connected to the problem of trying to maybe publish less and better papers than trying to pay reviewers so that they review these millions of papers. And for the anonymity, I think so first thing, if you discuss with editors, the first thing we tell you is that you never actually can guess who are the reviewers of your papers. And every time an author comes up to them and say, "Oh, I know, this was this guy," and they're always wrong.
No. If I may interrupt, I meant something else. So not you don't know the reviewers, but the reviewer that review your work, they might
you
know.
Mean anonymity of the submission. Okay.
Sorry,
I thought you were because there's also a discussion about should reviewers sign their reviews and you know who they are. And the thing is, from the discussion I had with editors, they're always saying, the people you think are your friends are not your friends, and the people you think are your enemies are usually much friendlier than you think when they review your papers. So I just don't play that game. Like when I have a student say, "Oh, this review might be for," don't even start. Don't even try. So, but anonymity of the submission. I guess it's as you said, it's a human process, and if you are an expert in a domain, I have difficulty to think that you cannot know where a paper comes from just by looking at what is done and how it's done. So I don't think it's what I think is very helpful to get a bit more objective review is to have a collective what eLife has been doing, which is you have a collective consultation of the reviewer, and they all discuss together to have a single review that reflects all the reviewers. Because it kind of irons out, the things that would be completely out of step, or the things that are just stupid, or the things that comes from misunderstanding from one of the reviewers. So you get a review that is much better, that is much more anonymous actually, because it's very difficult to know which part comes from who, and so I think this is I'm a big proponent of having it's more work for the editor, it's more work for the reviewers, because you have this step where you have to put everything together. But I think it's a way to have something more objective. And as regards the way, the fact that you know who you are reviewing and the authors are not anonymous, there's not much you can do about this, and we know it's proven by actual academic work that it has a role, and that if your paper comes from a very reputable lab, it will be easier to go to a good journal than in a completely unknown lab. And this is, for me, one of the dangers of the new landscape of publication. We say, okay, we don't want to look at the name of journals. We want to be completely free of those bias, and we want to just assess the quality of the work. But when you ask people what is exactly the quality of the work, and usually what comes out afterwards is, it comes from that lab, so it's good. So it's like you are replacing a system of impact factors and things that are completely wrong, but you are replacing it with a system that is just, I know these people, so they are good, which is not always better, so we have to be careful about that, and to not replace what we think are biased by something that is also biased, which would be like pedigree and where you come from to decide what is your value.
I think this is a great great opinion on this problem. There is also something that bothers me a lot, is like, if you want to submit to one journal, let's say you worked for 3-4 years on some project, and it's very nice data and the story is beautiful, and you want to try submitting it to one of these big journals, right? And then eventually it gets rejected for whatever reason. You want to submit it to the other one, you have to reformat everything in case of like text and pictures to submit it somewhere else, and which is also very, very...
So I have an insider secret for you. You don't need to do that, actually.
Okay.
Most of the time.
We can talk off screen.
No, I mean, I remember when I was a PhD student and I would read the author guidelines, that it was the gospel, and trying to get the lengths exactly and the presentation. And actually, for a first submission to a journal, before it's even reviewed, they don't care. They will ask you to change everything if they want the paper after revision. So of course you don't send a paper that is 3 times the length that is published in that particular journal. But if you have a bit above, or the figures have little a's instead of capital A's, that doesn't matter. You will change that if you are revising it and then you see it and the journal wants it. They will bother you. Don't worry when you are much later in the process, so that everything is like they want, because their goal is that you do as much of the work they should do yourself. So they will ask you to do it, but you usually, and a lot of journals now actually state that very plainly, like, first submission, send us just a PDF, whatever formatting you want, and then later on, if we really want it, we will tell us to make it exactly to the format. So that's something. And the other thing is that you have now new schemes like review commons, which I think are very interesting. Like you are decoupling the review from the journal. So I don't know if you're familiar, but review commons is operated by MBExC. And the idea is that you publish your preprint and then you send it to review commons, and they arrange the reviewing by 3 reviewers exactly like a journal, but there's no journal at this step. Like it's just they are reviewing the science and saying this is good, this is bad. I think to be an exceptional work, you should really do this and this and this and this and this. And once you have the reviews, and you make a review, you make a plan for revision, and then you submit to one of the journals of the Review Common Consortium. Like there are, I think, 28 now, and mostly the most famous ones are cell biology journals, like Journal of Cell Biology or EMBO Journal. But basically, you submit your paper, the reviews, and your revision plan, and the editor within 10 days tells you, okay, if you do this revision plan as you state, we will consider it as a revised manuscript. So basically, we will consider accepting it after sending it back to the original reviewers. And 10 days there, and the editor say, no, we are not interested. You can go to another journal of the consortium, and it's only 10 days. So you can really quickly find a journal that is really interested. And the other advantage is that the reviews are not about the fit or the level of the journal, because when the reviewers are reviewing, they don't know where you will submit it.
Yeah, that is actually, I was not aware of this, but I will definitely
I hope
read
it will catch on in Neuroscience, and that more neuroscience journal will be part of that kind of initiatives.
Yeah, let's hope for that. We are almost 1.5 hours in, and you're going to travel now to Frankfurt. So I would love to talk to you much more, but we will not keep you. Hopefully, you're not gonna miss the flight. So thank you very much once again for coming and for talking with us about these exciting topics. And maybe see you around to one of the conferences, or come to Marseille and visit the lab. I don't know.
Yeah, I'll be very happy to be back, and if any of you guys are in Marseille, I'll be happy to have you for a visit.
Yeah, thank you very much.